Ladies and gentlemen, please welcome Genome BC's President and CEO, Suzanne Gill.
Hello and welcome. Thank you for joining us for the annual Don Ricks Distinguished Keynote Address. I know you are all excited to be here. This is the 15th year of this incredible event. My name, as you know, is Suzanne Gill, and I'm the President and CEO of Genome BC. Before we begin, I would like to acknowledge that the land on which we gather today is located on the unceded territory of the Coast Salish people, the Musqueam, Squamish, and Tsleil-Waututh nations. This land acknowledgement is one way we could recognize Indigenous presence and land rights and recognize colonial history and the need for change. It is a thrill to look out and see this room full of curious minds, and thank you to all of you for joining us. And we're in for an exciting talk with the Dr. Sam Aparicio in a few minutes. We are also honored to have a very large, about a quarter of the room here is full of students. So special welcome for those high school students from Mosscrop, Gladstone, and Heritage Wood Secondary, and to members of Science World's Future Science Leaders Program. You are really the next generation of researchers, clinicians, innovators, so we look forward to you continuing your studies, and I know you had a good chat with Dr. Aparicio a few moments ago. I would also like to take a moment to acknowledge and remember the person this event is named after. Don Ricks was a visionary and a philanthropist who left an incredible legacy on the Canadian life sciences sector. His passion for advancing healthcare led him to establish Life Labs, a network of clinical laboratories across Canada. He was also the founder of Genome British Columbia. He believed in the transformative power of technology and innovation. Don was also a cancer patient. He was the first patient in BC Cancer's bold personalized oncogenomics program. And he was the first person in the world to have their cancer tumor sequenced. This was back in 2010. And this helped his doctors personalize his cancer care, his treatment, and the drugs he received from his clinical care team. He really was POG, as Mark O'Mara reminded me a few moments ago. And that sequencing effort was really possible thanks to world-class genomics research, the BC Cancer Foundation, and the scientists, some of which are here tonight, Steve Jones, Mark O'Mara, Janessa Laskin, among others. Truly, in all facets of his life, Don was a pioneer, and we are very privileged to have Lori, his daughter, in attendance with us today. Lori, thank you so much for being here with us at this event named after your father's legacy. And we are grateful for Don's leadership in biomedical research and life sciences, and his commitment to innovation inspires us to push the bounds of scientific discovery. Genome BC was founded nearly 25 years ago to harness the promise of transformative medicine, and that was to come from the completion of the Human Genome Mapping Project, $3 billion, 13 years of effort. 10 years later, in 2010, Don's cancer was sequenced for $250,000 or thereabouts. Today, cost is several $1,000s. Imagine what will come with technology advances and the costs coming down. Over the past 2 decades, Genome BC has invested in world-class research, supported innovative companies, and forged strong relationships with industry, academia, and our Indigenous communities. And through our work, we've helped build a strong, collaborative, globally recognized research and innovation ecosystem. And evidence of this is not only in the outsized impact of our research, but the incredible life sciences sector and industry growth we see in BC. And I see Wendy in the audience. We are the fastest growing life science sector in this country. Some examples of Genome BC's research and impact include the development of a test for hereditary cancer that reduced test result wait times from over 1 year down to 3 weeks, improving the surveillance for emerging public health threats, pathogens of concern, if you like, by using genomics to monitor wastewater systems in our city. In forestry, the application of genomics for climate-adaptive breeding so the province and industry can plant trees that will have resilience to warmer temperatures and drought conditions. And in resource development, our mining microbiome project is collecting and analyzing environmental samples from mine sites to identify microbial communities that we can look at to replace chemicals used in mineral extraction and in the remediation of mine sites. Now, just a few things before I turn it over to Federica about today's program before we introduce our speaker. Following the presentation, there'll be a Q&A for about 30 minutes, and you can use the microphones that are in both of the aisles or Slido. And today's conversation isn't happening just in this room, so yes, all those students out there, you can be on your phones for this. The team will be on X, Instagram, Facebook, and LinkedIn. And after today's talk, we invite you to complete the survey. This event is for you, so if you have feedback for us, we'd love to hear it, so we can do a great job next year. And you will also see the green lanyards in the room. Those are Genome BC staff. We're a really friendly bunch, so go find us and talk to us afterwards. And this year, we're also delighted you will have seen a number of booths outside in the reception area. Our partner organizations in cancer prevention, care, and treatment, Canadian Blood Services, the Inspire Health Cancer Care Centre, and the First Nations Health Authority. And one of my favorite, there's a pop-up Gene School booth, which you have to go visit, and there's a trivia challenge. So please enjoy that with, I guess you scan the QR codes and go find your answers. So for everyone here today, this event is for you. Have a good experience, and I would like to ask Dr. Federico De Palma up on stage to introduce our speaker.
Thank you, Suzanne, and welcome everyone. So today's guest obviously follows in the best traditions of our Don Ricks Distinguished Lecture. Dr. Sam Aparicio is a teacher, an innovator, and a leader in BC healthcare and cancer research fields. Dr. Aparicio has a remarkable career and pioneered important discoveries in the field of cancer genomics. He trained in internal medicine in the UK at Cambridge and Oxford universities. He earned degrees in medical and natural sciences, as well as clinical medicine. He followed all of this with a PhD at Cambridge, where he worked on the genetics and genomics of human disease under the supervision of the Nobel Prize-winning geneticist, Dr. Sidney Brenner. While at Cambridge, Dr. Aparicio led a research group and co-founded a biotech company. In 2005, he moved, lucky for us, to UBC and BC Cancer as the Nan and Lorraine Robertson Chair in Breast Cancer Research. He is a distinguished scientist and the head of the Department of Breast and Molecular Oncology at BC Cancer. He is also a professor in the Department of Pathology and Laboratory Medicine at UBC, as well as a UBC Distinguished University Scholar. Dr. Aparicio is also the Canada Research Chair in Molecular Oncology, and finally, a Fellow of the Royal Society of Canada, which is Canada's highest scientific honor. His academic career has been nothing short of extraordinary. It's marked by groundbreaking research and significant contribution to our understanding of cancer biology that have been published in many high-impact journals. He led breakthrough work decoding the genetic makeup of the most deadly form of cancer, known as the triple-negative subtype. His work is changing the way the breast cancer is diagnosed. In today's keynote address, titled Decoding Cancer, Dr. Aparicio will share insight into how recent breakthroughs in genome sequencing have reshaped our understanding of cancer. He will highlight how British Columbia is making a vital contribution to this advancement and discuss our ongoing challenges with an outlook for the future. Dr. Aparicio's career has been really committed to integrating in his research cutting-edge technology with an understanding of cancer to prevent and diagnose cancer. So, ladies and gentlemen, I'm very pleased to introduce Dr. Aparicio today to deliver Genome BC 2025 Don Ricks Distinguished Keynote Address. Welcome, Sam.
So thank you all for being here tonight. I'm very honored to have been asked to give this keynote address today. We've been living through a biomedical revolution in the last 2 decades for anyone who works in the cancer field or indeed in the genetics of disease. Our understanding has been completely transformed by some of the science and technology, which I'm going to describe in a brief way in my talk today. But before we go there, I'm going to maybe take a step back and talk about how we got to decoding the genomes of cancer, which is something that I never thought I would witness in my professional lifetime, and to explain some of the impact that that has had for our understanding of cancer. Now, in Canada, 1 in 2 Canadians or their families will be affected by cancer during their lifetime. It's a common disease. But that wasn't the case back in, say, 16th century, 17th century England. And this diagram here comes from, it was called a bill of deaths. So people were recording deaths and beginning to do epidemiology on the causes of mortality for many hundreds of years ago. And if you read the Old English here, what you can see, this bill of death was drawn up in 1665, when the average life expectancy was around 36 years of age. And the primary mode of death in that year, 2/3 of the recorded deaths were from the plague. Cancer doesn't really appear on that list. There are many other things that were noted, including 23 people who died from fright, whatever that was. Okay. But cancer was not really a prime factor among them. Now, that changed as our societies learned how to separate the water we drink from the water we excrete into, and to have security over our food supplies and the way that communicable and transmissible diseases work. And so this summarization here from looks back to 1900, when still infectious diseases were making quite a strong pitch in terms of their impact for mortality. Tuberculosis, pneumonia, the Spanish flu was around in that period, gastrointestinal infections, but heart disease was beginning to be recognized as a cause of death. Cancer still not up there. By 2010, that's completely changed. So our societies, our modern societies, having eliminated or managed most of those threats, now people are living longer, and as average life expectancy increases, so the risk of developing a cancer and suffering the consequences also increases. So cancer is not exclusively a disease of aging, but the first biological clue as to the nature of cancer is that it is much more prevalent in older individuals. And to lead you on the journey here about what we've learned about cancer genomics, I'll just begin by making a very simple statement, which is that cancer is, in fact, it turns out, a disease of the genome. The instructions that are stored in our cells that make up our tissues become corrupted over time. And this partly explains the age dependency. It's partly due to a defective copying of those instructions over time that lead to cancer. But this says nothing about how and where are the actual building instructions for a human cell stored and maintained. And so I want to take you a little bit on that journey, because I think to understand what we've achieved in cancer, decoding cancer, you have to understand how did we get to the first human genome sequence. So this story goes back a little bit, and it starts in 1865. So in 1865, there was a fellow who I'm sure many in the room will have heard, high school science talks about genetics, and that's Gregor Mendel, the monk of Brno. He was a sort of biologist, mathematician, meteorologist, monk, kind of jack of all trades. And he was the first individual to really codify the idea that traits that we all recognize in individuals and plants and animals in all natural living systems can be inherited from one generation to the next. So this idea that physical traits that are passed on, which can be inherited, he captured in the form of mathematical rules. He was the first person, in a sense, to be the mathematical biology of hereditary, a subject that we now call genetics, which is the study of heritable traits. And it's worth noting that at the time Mendel formulated all of this, he knew nothing about DNA, he didn't know about chromosomes. He actually did know something about evolution, not about cancer evolution, but he was a devotee of Charles Darwin's works. The reciprocal is not the case. So Darwin actually knew nothing about Mendel. But it's known that Mendel was quite influenced by Darwin. And he published all this, so he wrote all this stuff down, and then it was forgotten about for 35 years, which is kind of like a scientific entire lifetime in a way. But time moved on, and around the turn of the 20th century, the dominant technology of the time was microscopy. And so microscopists were beginning to study how cells make up tissues. And here's this pictograph I have here is from a publication by a guy called Walter Fleming, who provided the first physical description of how cells divide and what goes on in a structure called the nucleus of the cells. And microscopy discovered from using the stains that come from the bark of trees and microscopes that there were these structures which were called chromosomes because they absorb pigments, chroma for pigment, and that chromosomes underwent this kind of these strange movements as cells split and divided. Wasn't really fully understood until around 1902, 1903, when there was a kind of a revival of Mendel's work. It was rediscovered by three individuals, William Sutton and the Bovarys, Theodore Bovary and Marcella Bovary, who were studying how cells divide in parasitic worms. And Sutton and the Bovarys were the first to posit the idea that these chromosomal structures might be the units in which, or the structures in which heredity might be based. An interesting side fact here is that actually some of this work was conducted by Marcella Bovary, who was married to Theodore, who supervised her husband's graduate students after his death, and it said she was much tougher on his students than he was. But the Bovarys in 1914 noticed that in tumors, physical tumors that were obtained from animals, that they could see abnormal chromosomes, so chromosomes that didn't appear in the usual way but had some kind of structural anomaly, like broken and so this was really the first idea that chromosomes might be somehow related to cancer. So genetics as a field then developed. One can ask, these units of heredity that became known as genes, how are they actually organized? And at first, no one really knew anything about the organizing principles, but it turns out that the genius of Mendel's mathematical description of the rules meant that it was possible to construct a thing called a map, a physical map. We're used to the idea of looking up where we are on the Earth by looking at a map, and it turns out that if you want to understand how genes are organized in a cell, you can also have the concept of a map. And biologists who were studying fruit flies around 1913 managed to start to make these conceptual maps. These are not real physical maps, but they tell us about the relative ordering of genes using the rules of heredity. And eventually the physical characteristics provided the anchors for the heritable units. Okay, but this still doesn't tell us what's the biochemical substance that's responsible for heredity. So that took a bit longer. And in fact, until 1952, it was widely believed that proteins were the basis, the transforming principle of heredity. And nucleic acids, which were known, biochemists had discovered these molecules made up of sugars and phosphates and purine pyrimidine bases called nucleic acids. There were only 4 distinct biochemical varieties of these, and no one could really imagine how you would store information about making up a complex organism or a protein from that kind of information. So that took a little longer, but into the picture came actually a Canadian, a guy called Oswald Avery, who was working at Rockefeller at the time, together with MacLeod and McCarty, and they showed, using experiments in bacteria, that the principal molecule that would transform the characteristics of a bacterium was not a protein, but DNA, and they managed to prove this. He was nominated for a Nobel Prize many times and was overlooked. It was subsequently the Nobel Committee sort of admitted this. But the people's thinking really changed after an experiment that was conducted in 1952 by Martha Chase and Al Hershey at Cold Spring Harbor. They used a blender, the kind of thing you might have in your kitchen for making a fruit smoothie. What does that have to do with DNA? Well, they managed to show, using radioactivity linked to proteins and DNA, that the thing that caused the transformation of bacteria with bacteriophages was in fact the DNA. And that really did change people's ideas. It became apparent that somehow nucleic acids were conveying heredity. Now, of course, the famous biochemical basis of genetics, that really was a subject transformed in 1953 when Francis Crick and James Watson, who were informed by data, X-ray crystal diffraction data that was generated by Rosalind Franklin and Maurice Wilkins, proposed a structural model for DNA, which explained how genetic information could be encoded and replicated. And the key features of the model were the idea that this information was stored in a way where the biochemical bases, the 4 biochemical bases of DNA that we now call, we use letters A, G, C, and T, they represent the different purines and pyrimidines. They have a certain base pairing relationship, which means that the linear order gives you potentially information about what's encoded, and the copying mechanism is enforced by the fact that only certain base pairs will pair with each other. And so that became immediately apparent from this model. But what nobody actually knew at the time, so it was obvious that there could be a code and how it might be encoded, and it was obvious how it might be copied, but nobody knew what was the code. So that took a little bit longer. And there was an experiment conducted in the early 1960s by Francis Crick and Sidney Brenner. Here's a picture of Sidney, by the way, who visited UBC a number of times with one of his last Cambridge PhD students. That's me. Okay, and Francis and Sidney showed in 1961 that the code was commaless, so it had that property. It must be a triplet. In other words, it had to be made up of 3 nucleotides, that it was non-overlapping, and it was read from a specific starting point. Those experiments were done without any modern knowledge of how to actually determine the sequence of nucleotides. It was all pure genetics. The code itself was actually decoded by 3 biochemists, one of whom has an important connection to BC, and that is Gobind Khorana. If you've ever wandered around the new Westbrook Village development in Point Grey, there's a little park there called Khorana Park, named after Gobind Khorana. And he was a nucleic acid chemist who started his career at UBC and pioneered nucleic acid chain synthesis. And then together with Marshall Nirenberg, who was a biochemist at NIH, and Robert Holley, who worked on tRNA, they literally worked out the code. So they worked out that certain sequences of nucleic acids would code for certain amino acids. And this is how a protein sequence was determined. So 1961 to 1966, there was a code. The code was known. But then the next question comes, okay, the code was known, how to read it? Now, to understand just how hard this is, you have to understand something about the dimensions of the problem. So one human cell was known to contain 23 pairs of chromosomes. The DNA, if the physical piece of DNA from any single cell, if you were to take it and stretch it physically, this is from something that's microscopic and you can't see, would stretch 2 meters, which is the equivalent of 40 kilometers of thread packed into a tennis ball, if you can imagine that. It's absolutely mind-blowing how all of this information is packed into such a small space. There are something like 37 trillion cells in a human body, which would be enough if you stretched it out to be 2 times the diameter of the solar system in each person. The linear sequence of one set of chromosomes contains about 3 billion bases, and of course most of our cells have 2 copies, so it's really 6 billion bases. So it's a phenomenally difficult problem to think about how to actually biochemically unwind all of that sequence structure. The first method for reading the order of DNA bases, which is what we mean when we say DNA sequencing, was invented by Fred Sanger and Wally Gilbert. It was actually Fred Sanger's 2nd Nobel Prize. He won an earlier Nobel Prize for having discovered the structure of insulin. And Sanger's first method of sequencing used a biochemical approach called chain termination. It's a method that depended on physical separation of DNA chains differing in length by 1 nucleotide unit. And there's a picture here which kind of conceptualizes how this was done. What Sanger, who was a biochemist, managed to do was to develop nucleotides which, instead of being routinely incorporated into an elongating chain of DNA, there are enzymes which were discovered from cells that will copy DNA strands and allow a single strand of DNA to be extended with its base pairing. He discovered nucleotides that are called chain terminators that have the property that when they're incorporated, they stop further incorporation. And it's possible to set up a biochemical reaction with chain terminators for guanine, adenine, thymine, and cytosine separately, and then resolve. What that does is it produces a ladder. So every time there's an A or a T or a C in the sequence, it will stop at that point and incorporate a little piece of radioactivity, which is how the ladder pattern is produced. And this method of chain termination was the first method of sequencing. I started my PhD working with this kind of sequencing method. It's really laborious. You could sequence fragments of around 300 base units, and the organization of gene coding sequences could then start to be recognized, but only fragments were recognizable because this was such a slow and painful process. But at this point, it became theoretically possible. It's a thing that scientists sometimes like to say, in principle. In principle could mean with $3 billion and a lot of effort. But in principle, we could decode the sequence of a human genome. Now, that was in fact launched in 1990. And without going fully into the history, I want to say that this was about the time I was starting my PhD. The Genome Project was launched with considerable opposition. There were a lot of letters written and a lot of energy expended saying we shouldn't do this, it was going to be too expensive, and so on and so on. And the way it was done was a combination of automation, scale, and computational power. So the Sanger method, that basic Sanger method, was to an extent automated, and this automation, if scaled over enough countries and enough instruments, and literally the genome centers of the time had huge rooms, the size of this room or bigger, stuffed full of these machines, reading out bases in the way that the Sanger chemistry dictates, spread over 6 countries, 20 centers, and thousands of researchers, reading about 1,000 bases a second for a long period of time. Brute force Sanger DNA sequencing allowed the first human genome to be sequenced and assembled. And it was a long road. This graphic put together by the NHGRI sort of exemplifies many of the steps. I've told you some of the earlier ones, which led to the idea that there is a code, the code could be read, this was the biochemistry, and then this is how it was done. The first draft sequence was 2001. The completed first draft was declared in 2003. So in 2003, the project's declared victory. And we are the first species on this planet to have decoded the genetic makeup of our own cells and of other organisms. It was a profound moment. And British Columbia was not behind in any way. So here enters Michael Smith. So Michael Smith, who worked with Gobind Khorana and won a Nobel in '93 for his work on site-directed mutagenesis, was prescient in, together with the research leadership of the time, recognizing that Canada and British Columbia should establish a genome center, and that was indeed established. The founding director and Mike Smith was the founding director and recruited Mark O'Mara and Steve Jones, who led the center since its inception in 1999. And I'm grateful to Marco for sharing these pictures. It was the only center that was it was the only center globally that I'm aware of that was also co-located within a cancer center, and that's the BC Cancer Agency. And the relevance here is that one of the rationales for the Genome Project was originally that cancer is a disease of the genome. So why study the human genome? To get at cancer. Now, the Genome Center has, over its lifetime, made many, many contributions. It put Canada on the international genomics map. This started with contributions to the physical map of the human genome, and then the initial sequencing and analysis of the human genome. The SARS-1 virus, which some of us will remember, decoded in 2003. That was the first brush with SARS. The Genome Sciences Center established itself as a leading edge technology development center. It established the computational infrastructure and methods development that are necessary. It trained an awful lot of people. As of 2019, I think over $1 billion worth of projects, 96 different funders, something like 2,000 people employed, of which around 350 at any one time. 95% of it funded from external grants. So a remarkable organization with global impact. But the thing that is relevant to know here is when the first human genome sequence was completed in 2003, decoding cancer at scale was not immediately tractable. The first genome took 15 years and cost around $1 billion to $3 billion, depending on how you cost it. Circa 2008, the Sanger technology, which has been the basis for all the sequencing, was still pegged at somewhere around $37 million for one haploid genome. So not $1 billion, but far more than anyone would want to think about for undertaking research or diagnostics. And so the automation of the Sanger method had pushed the cost down and increased the scale, but the hard floor limit was intrinsic to the technology. You just couldn't get any further. Now that changed, and I'm going to explain how. But the scale of the problem in cancer is even larger than just saying we want to decode another genome. Mutation happens in cancer. I stated that at the outset, that cancer is a disease when DNA gets copied imperfectly in different ways. And it happens at all scales of the genome, from single bases, like it could be a single letter, one letter in the 3 billion or 6 billion letters. It could be cut and paste segments of the DNA, which lead to changes in the gene dosage. So you might have 3 copies of a gene rather than 2, all the way up to having duplications of the entire genome. It might be shuffling of the structure of chromosomes. On the right here is a thing called a spectral karyotype. I'm grateful to my colleague David Huntsman for this picture. Normal 23 pairs of chromosomes painted on the top. You can visualize with a microscope. The thing on the bottom is a cancer that's gone completely wild. It's lost control over the number of chromosomes. They're shuffled all over the place. So it's not simply that you have to sequence the DNA. The DNA is different. It's mutated. The other aspect to the problem has to do with Charles Darwin, who's basically in the driving seat. Evolution is a property of all replicating systems. Anything that can be copied and propagated has the capacity for the copying to be imperfect. Mutation is the substrate for evolution. And Charles Darwin knew nothing of DNA or chromosomes, but he codified these principles when understanding how species originate on Earth. And it turns out that Darwinian principles operate in cancer, where ongoing mutation means that cell division and mutation means that every cell in a cancer could in principle be different from every other, and that mutation creates a subspecies of cells. We refer to them as clones, which means that cancers can evolve under treatment. It's a fundamental property of cancers that they change over time and treatment. And that's something that we've been studied. So a complete understanding of this, if you want to get to grips with this phenomenon, you have to decode the full sequence of corrupted cancer genomes, and eventually every single genome of a cancer. Is that even possible? I didn't think it was possible. But in 2006-07, there was a revolution in DNA sequencing which took place, and it has completely transformed not only cancer biology, but many other aspects of human medicine. And I'm grateful to my Cambridge colleague, Shankar Balasubramanian, who invented this technology for the figures I'm going to show. And there were really 3 things that had to happen. The first, and the question was how to decouple the sequencing process from the physical separation of DNA molecules, because that was really rate limiting. So the first thing, and this was developed around 1995, was a way of visualizing single DNA molecules actually being extended as a chain with an enzyme. And that was done with fluorescent nucleotides that could be incorporated and clever microscopy. So that's the first thing. The second thing was to make different kinds of terminators. The problem with the Sanger reaction is that once the base is incorporated, you can't extend the chain anymore. And what you really want to be able to do is to sequence by synthesis, where you repeat the process after reversing the cycle. And so Shankar developed chemically reversible terminator nucleotides that could be incorporated, read, and then their termination function could be eliminated chemically so that the chain could be further extended. The third part of this was the scale comes from the ability to physically array single DNA molecules on a surface, so that you take an extract of DNA, you put it on effectively a microscope slide, you get single molecules, and then you can repeat these cycles. And this leads to a picture. So now it's sequencing by microscopy. The microscopes are back in the modern generation, and the picture on the bottom right there shows a fluorescence image of a surface where these cycles of addition and extension have been repeated, and those colored dots will flash on and off in different colors depending on the base that's being incorporated. But now these molecules are arrayed over a vast entire physical surface. There are billions of them. Suddenly the sequencing is massively parallel. And this was developed by a company called Solexa in Cambridge, which was eventually acquired by Illumina. This is now the technology that is used in 90% of the world's DNA sequencing. The Gairdner Foundation recognized their contributions this year. There was a prize given to Shankar, David Kleineman, and Pascal Meyer for the invention of next-generation short-read sequencing. And it's not an exaggeration to say that when this moment arrived, the floodgates opened. And BC was in the fortunate position of being only one of 4 centers internationally to have a Solexa instrument. I'm grateful to Marco for this picture of the device. This is the first one of its kind, went through many evolutions. And as we know, what this did was it shoved the cost of obtaining this genomic information down by orders of magnitude, and in fact outpaced a thing called Moore's law, which is something in the computing field that explains the growth of doubling in computing power every so many years. The cost fell dramatically, currently around $300 for an equivalent genome information. The scale and the throughput rose so that a genome could be decoded within weeks to days. And the sequencing technology was single molecule, which opened up the study of cancer evolution. So we were really fortunate to be in this position. And what happened subsequently in the cancer field is that there was a vast explosion of effort to start to decode systematically cancers. There were a number of international projects, some US-led, looking at different tumor types. Another set of groups were the International Cancer Genome Consortium. But the great thing about the technology is that it essentially also democratized a lot of the sequencing and meant that individual centers and labs could contribute. And BC made many contributions. The very first cancer genome to be decoded by this method was a blood cancer, an acute myeloid leukemia. That was done by Elaine Mardis and Tim Lay at WashU. They established a key research paradigm, which is that in order to interpret these cancer genomes, we have to have a source of normal DNA because there are many private variants that you have to eliminate from the sequence. The first solid cancer genome came from our center. I was quoted at the time as saying I never thought I would see it in my professional lifetime, and I didn't. We were fortunate enough to be able to take a lobular breast cancer that was resected around 2000 but recurred in an individual patient as a pleural effusion and metastasis, and we were able to sequence the genomes of these two situations and compare the early cancer with the evolved cancer. It's the first time anyone had looked at cancer evolution this way. And the impact of the mutations in the gene expression could be seen for the first time. We measured cancer evolution. the first of many studies with my colleague in medical oncology, Karen Gelman, and my good friend and colleague, Saurabh Shah, who's a computer scientist currently at Memorial Sloan Kettering, but we did many of these studies together. Cancer evolution has since been decoded in many studies. We've understood how primary and metastatic cancers spread. We've understood that cancers have different regional compositions in mutations. If you sample one part of a cancer and a second part of cancer that are separated in space, you may end up with a different idea about the mutation content. We've learned about the origins of chemotherapeutic resistance. We've learned how the immune system might interact with clones. So much has been learned about cancer. Another world first from BC, it's been mentioned in the introduction, was the first decoding of a patient's genome with a therapeutic management intent. This paper is now famous. It's been quoted many times as the first example of a personalized oncogenomics approach to clinical medicine. And Marco and Janessa and many colleagues at BC Cancer created the BC Cancer Personalized Oncogenomics Program, which has helped many patients with cancers that are beyond standard of care options in terms of helping with management, and really was a paradigm for many other such programs around the world. BC discovered many new cancer-causing genes. There's a selection here. Ryan Moran, working with Marco and the lymphoid cancer group, discovered cancer-causing mutations in a special type of lymphoma, and we later worked out the biochemical mechanism by which those mutations cause the cancer. My colleague David Huntsman and Ovcare discovered mutations in a gene which is called ARID1A. The names are never very interesting in these genes. There was a subgenre in genetics for naming genes after interesting things, but it ran foul of copyright laws. The Pokemon causes cancer led to some lawsuits. Okay. ARID1A is the second most frequently mutated gene in cancer, it turns out, a really important discovery. And it was made here in BC, sort of co-discovered with a group in the US, but this was among a flood of new discoveries. Working in breast cancer, we, together with colleagues in Cambridge, we led a study that was published in 2012, which looked at a huge number of breast cancers that we were able to study because of the large clinical repertoire of cancers available at BC Cancer. We discovered that there were 10 subgroups of cancer. We learned about the impact of gene dosage mutations. We could see that metastatic breast cancer is very strongly influenced by gene dosage mutations. The BBC thought that was the top medical breakthrough of 2012. Well, who knows? But this is another feature of the cancer genomics landscape, is that all of this activity has been pushed into the public domain. We all push our data sets out. Everyone can use them. There's been huge reuse of data. This particular study has been cited over 6,000 times and used by 600 groups. There are many other examples. This has accelerated progress by having access to data. In 2012, a much younger Martin Hurst and Stephen Cheer and my colleagues at BC Cancer, we were the first to decode the genomes of triple-negative breast cancers, which is a particularly difficult to treat heterogeneous group of patients. We've learned since in more recent work, using more modern genomics, that triple-negative breast cancers and ovarian cancers are actually in some ways linked. They look like they occur in different sites of the body, but the way in which DNA gets copied and shuffled in these diseases turns out to be the same. This graphic shows you a summary of some of the different ways in which copy-paste operations that are mutations can lead to duplications of genes or deletions of genes, or indeed folding back of chromosomes onto each other. And it turns out that these patterns explain different underlying mutational processes that are abnormal in the cancers and which seem to be associated with different responses to chemotherapeutic drugs. We don't fully understand why that is, but we now have a biomarker with which we can use to identify these patients and study the problem. The idea that cancers are different from each other, this is another fundamental aspect of cancer that's been addressed by the kind of modern cancer genomics. Every cell in a cancer can, in principle, differ from every other cell, because every time a cell divides, there are a few hundred to a few thousand mutational variants that are created by the copying process that are not corrected. So that means if I took every one of your cells and I sequenced them absolutely end to end, I would find a few hundred differences at least between every single cell. So this mosaic that you see here represents the idea that a tumor at the time of initiation is composed of different groups of cells. We call them clones. At diagnosis, it may have evolved some more because some of these clones may have greater capacity to survive than others. That's called fitness. Indeed, when cancers relapse, we often find that it's a completely different clone, shown in green at the end here. So this process leads to cancer evolution and drug resistance. And if you doubt what that looks like in the clinic, this rather dramatic clinical example shows evolution in action. This is an individual who had a metastatic melanoma. That's the picture in A on the left. You can see that there are little melanoma deposits all over this individual's skin. They were treated with a drug that treats the mutation, the driving mutation, and magically the cancer's gone after 15 weeks, or so it appears. But unfortunately, at 26 weeks in this individual, they all came back. Why? Because the cancer cells have mutated a resistant form that is now resistant to the drug. So that's cancer evolution in action. And it's one of the biggest problems that's faced in oncology is that the drugs, if not fully successful, will eventually lead to resistance. Now, this leads to another area of genomics, which is called single-cell genomics. The idea that every cell is different and location matters. It's a bit like, you can think about the analogy here. It's the fruit smoothie analogy, okay? So if you have a bunch of fruits that are going in to make your fruit smoothie, what we've been doing in genomics is the equivalent of the thing on the far left there, which is putting everything literally in a blender, which destroys all information about what fruits were in there and where they were located. Now we can read single cells, so we can read them in a dissociated form, like the picture in the center. We can tell what fruits were present. And with spatial single-cell genomics, we can tell what their location was. And so all of this information matters in terms of trying to understand the biology. We developed a technology for this to sequence individual genomes of cancer. It was developed here at BC Cancer, and it's been exported to many centers now. We've sequenced something like 10 million single genomes. So I started this by saying the first genome took $1 billion in 15 years. Now we've decoded 10 million genomes in the space of just 2 or 3 years. The technology has completely changed, and we can begin to see that, as you can see in the graphic in the center, these are 3 genomes from 3 different cancer cells. And you don't really have to sort of appreciate the details of this to see that every one of those cells is different. Every cell is different from every other. And so we can now study this process with genome sequencing. How different can they be? This is a portrait of a cancer. We're now studying Terry Fox's tumor. Terry Fox had an osteosarcoma. This is a portrait of an osteosarcoma. This heat map portrait, there are 300 genomes represented on here. Every line on this multicolored heat map represents a single genome that was decoded. And the gene dosage of each chromosome is shown along the horizontal. And you can see that on the bottom right there is an example of a single cancer cell. And you can see just how crazy the thing is. It's got lots of regions where there are gains in genes and losses in genes. Normal would be gray. It would just be 2 copies. And so you can see what this really means. Every cell is really different from every other. And so that's a rather profound thing to think about when you're thinking about cancer biology. But we've been able to exploit this to learn how clones behave under treatment. One example is a study we published just a couple of years ago looking at the common chemotherapeutic agent, platinum. And when tumor cells are treated with platinum, if you treat for long enough, eventually you end up in a situation where only one clone is selected. And if you take the treatment away, the cells, and then bring it back, the cells are still resistant to the drug. It's called fixed resistance. But we discovered that in early platinum resistance, if there are just a few surviving cells that are not affected by the resistance mutations and you take the treatment away, often they just grow back. And what that means is that there's a cost to a cell, there's a genetic cost to a cell becoming drug resistant. The resistant state is in some ways paradoxically weaker than the non-resistant state in the absence of drug. And this is a profound phenomenon that I think is teaching us about cancer biology. Another, I think, very important output of the programs here has been the development of computational methods. It would take me a whole lecture to go through the things that I've learned. I've worked closely with very many members of the Genome Sciences Center with my colleague Saurabh Shah and Alex Bouchard, who are in stats and computer science at UBC. There were many students who came through the program who either did their postdocs or PhDs through the program, who went on to training internationally, and some of whom have come back. Andy Roth, Dee Steef, Jerry Ding, they're now all UBC-based. We've recruited other members, such as Yongjin Park, who's ex-MIT, now at UBC, and stats at BC Cancer. So computation and development of computational methods is another thing that's been stimulated here, and I'm not doing it justice in this brief slide, but it underpins everything. So I've told you something about what's happened in the last 20 years, and I just want to sort of conclude in the last few minutes by saying what lies ahead. And we're beginning to look at questions such as the origins of cancer, the way that we might detect and monitor cancer burden, and how we might actually treat cancers with genome-targeting drugs. The first of these is a study which is actually going to appear in press this week in one of the scientific journals. Using the single-cell sequencing technology, we started to look at individuals without cancer and ask how early can we find the mutations. And it turns out that you can find a few mutations in everybody because we all generate mutations as our cells divide. It does not mean that the cells are tumors. Many of these cells will never divide again, and they require additional events to become transformed. But one of the things that we realized in looking at breast tissue with this method is that the mutations that seem to be the ones associated with eventual cancers only seem to arise in one type of cell, the milk-secreting cells, and not the other type of cell. And we have no idea why that's the case, but we've observed it. And so now we know that that's the study cell population of interest. Detecting cancer burden is something that can now be done from a genomics test. Malignant cells shed DNA into the circulation. It's possible to analyze that by literally taking a blood sample and putting it into a DNA sequencer. We recently conducted a study of this in triple-negative breast cancer, where what's shown on the diagram here is the anticipation in anticipating a clinical relapse by taking a blood sample. So the green dots here represent a blood sample, and the red-orange dots represent a relapse. And what we were able to see is that we could detect the seeds of relapse, eventual relapse, up to 27 months before an individual might have relapsed with a clinical cancer. So this has great promise for both detection and management of cancer, something that's being actively looked into. And the third area that we're actively interested in is the idea of therapeutics, and this is work that I'm doing together with my colleague Shankar Balasubramanian of Solexa fame. Shankar's other interest is folded DNA, and some years ago, around 2016, in my lab, we discovered a small molecule which turns out to have the property of sitting on DNA molecules that fold up into four-stranded structures. The Watson and Crick structure that I described earlier is a two-stranded helix. That's how most DNA is packaged. But it's possible for DNA to form four-stranded helices that fold up into little blobs in the nucleus. They're called G quadruplexes. And stabilizing these molecules with G quadruplexes presents challenges to a cell that has deficient DNA repair. And we've been able to exploit that through a clinical trial. We found a drug that had this activity. We were able to repurpose it. Together with the Canadian Clinical Trials Group, we published a study last year, a clinical study showing that this kind of drug treatment has potential merit. We're looking into this as a way of treating DNA repair deficient cancers. So I think just in closing, because we're down to the last minute or two here, I just want to, this is a bit of a list, but people say, what did we learn from the last 15, 20 years of large-scale cancer genomics? And the answer is an awful lot. We've learned that there are vastly more types of cell function involved in causing cancer, that the effects of gene mutations are not the same in every tissue, that most cancers require multiple mutations, that evolution, we're beginning to decode it and figure out how cancers are resistant to therapy. Some things I haven't talked about, the microbiome turns out to influence cancer risk and progression. We're beginning to learn something about which mutations might drive immune reactions. And we know that patterns of DNA breakage can teach us about the pathology of mutation. In diagnostics and treatment, we know that there are cancer drugs that are designed to be mutation-specific that are already in use. So these are products from the genome projects that have already made their way into therapeutics. Mutations used to predict the activity of drugs, identification of high-risk individuals, identification of environmental carcinogens, monitoring and detection, personalization of cancer vaccines. It's a long list, and the story is not complete. There are things we don't know about modifications of DNA bases that we can't read. We don't yet know how to predict. I'm saying this because if there are those of you out there who are thinking about research careers that might think it's all over, it ain't all over. Okay, I used to think that. It's not true. Okay, there's chemical modifications of DNA bases we can't read. We don't know how to predict which cancer mutations affect the immune system. We don't know how cells form a memory of their type and how that modifies cancer. We're just beginning. The environmental carcinogens are interesting, but we're just at the beginning of that story. There are new technologies for editing and rewriting genomes that are leading to therapeutics. CAR T cells are already in use. There are more surely coming. Genome risk stratification. So these are all great research areas to be involved in. And so a lot still to do. I want to acknowledge this journey. It's been an incredible journey, and I've been privileged to be on the road. Many patients for donating time, tissues, medical records without whom the research doesn't proceed. Many local funders. I want to highlight the BC Cancer Foundation, who have generously supported us for many years. Provincial matching contributions from BCKDF and Genome BC. Many national funders, many international funders that we get a lot of international funding. And my many clinical colleagues and collaborators, I haven't been able to mention them all, but all of us have traveled the road. I just want to part with this one last thought, which is that fundamental discovery in science is the engine of progress in medicine, and don't let anyone tell you otherwise. I'll stop there.
Thank you, Sam, for this wonderful, inspiring journey through cancer genomics. So we are now in our Q&A session. So I just have a few reminders. You may use one of the microphones which are located in the aisle to ask your questions. You can also use a Slido, and we should have directions on how to do that on the screen right now. You can participate in social conversation on X and Instagram by publishing a post using the hashtag DRDK2024. How do I do this now? See, I'll go down. You stay here.
Okay.
And I guess I'm going to start off. I don't have a microphone, so I have to go back there. Okay. Sorry. I'll start off. Oh, there are already some questions, but I have one that I wanted to ask you. This was such an inspiring talk, and you have taken us through this transformative journey, pointing out all of how the technology has really changed and allowed us to make all these important step forwards in the diagnosis and treatment of cancer. What, in your opinion, is the next frontier in the next decade for cancer genomics, and how scientists and public alike can help get us there?
Yeah, it's a big question, actually, and it's a hard one to answer because I don't have a crystal ball. Okay? No. But what would I think about this problem? I think we've learned quite a lot about reading genomes, and there's a lot more yet to learn from reading genomes. That is, decoding sequences, which we now do very cost effectively, and it's having this impact that we can foresee maybe how early detection and risk management might get more help from genomics than we currently have. It's still a big public health issue, is how to actually screen and detect cancer early. Big problem. I think there's application there. We're just right at the beginning of learning how to rewrite genomes, so how to deliberately change the structure of a genome. And that has a number of possible applications, but in cancer, potentially for therapy, for immunotherapy, for boosting the immune system. I think these are things that are coming, but those developments are going to take, I think, still time.
Thank you. Okay, so is there anybody from the audience before I go to the Slido questions? The lights are very bright, so I can't see, but let us know if there are anybody who would like to ask questions. So, well, you can read, with AI becoming increasingly important, how do you see it being integrated into genomics and cancer research?
This is the AI question. Okay.
That always has to be one.
Well, I'm told that if you're raising money, it's AI, and if you're seriously interested in the science, it's machine learning. Okay. AI is already making its way into medicine, and I don't think cancer is any exception. And maybe the easiest place to see where it's being applied is in image recognition and pattern recognition, because modern machine learning methods are incredibly good at taking large complex data sets and looking for patterns. And that has a lot of application in medicine. For example, interpreting radiographic images, interpreting histology, which is currently done by human pathologists. The way that we look at genomes is influenced by machine learning algorithms. So we use this kind of technology all the time. Where will it eventually go? I think it could be controversial, maybe that at some point screening for symptoms will be conducted by an AI chatbot and not by a physician. That's a bit challenging for people to think about.
Yeah. Thank you. You didn't mention anything about cancer stem cell. Could you comment on the relevance? Cancer stem cells.
Cancer stem cells it's a field of cancer research that has posited an idea, which is that it's actually several ideas linked together. But one of the ideas is that mutations that we see in the large part of a cancer actually don't originate in many of the cells that we look at. They originate in stem cells that are present in our body. For example, in leukemias, you may see a lot of malignant white cells that are part of a leukemia. But if you actually go back into the origin of the leukemia, you can look back and see that the cell that's giving rise is actually a stem cell in the bone marrow. And those stem cells have important consequences for us, because through evolution, the normal function is to maintain our bodies, maintain tissues that rely on cell division. So if they become mutated and corrupted, that's a big deal. And so learning about that biology has been important. Yeah.
Thank you. So we have a question from the audience, please.
First, I'd like to thank you for your amazing talk. It's really good to see the history, particularly the Vancouver history. I'm a geneticist. I was here actually way back when Mark O'Mara gave his first talk to try to get the job here. I was also at the Amgen Research Institute in Toronto, where I had access to the Celera genome, which was the private Craig Venter genome that was sequenced at the time. And more recently, I've had my whole genome sequenced, and it's up on the web for anybody to download to study. I'm actually really interested in genome-wide association studies and looking at the people who don't get cancer or other diseases that live to long ages. Is there any work being done in BC looking at these people? Why are they resistant to developing cancers and other diseases?
Okay, so I guess your question is about GWAS and, okay, so genetic association studies. I'm not aware of, there may well be people in BC who are doing exactly what you describe. I'm not aware of that. What I am aware of is there are now many internationally based studies looking at GWAS because, as you know, one of the issues in looking for genetic associations is you have to look through an awful lot of people, individuals, because the association between variation that we find in our germline and the phenotypic effect that you're looking for is sufficiently weak for most of these effects that you really need to look at tens of thousands, if not hundreds of thousands of individuals. So that's really, I think, the subject of big international projects. And I know there are many researchers in BC who are members of those projects. Yeah.
Okay. Thank you.
Thank you. And over there now.
Is this working? Okay. Hi, Dr. Aparicio. I wanted to ask about one of your final culminating slides. You said how, like, we still don't know how cells know their type. And I was wondering if you could elaborate on that, whether it's clone type or specific cell type.
Yeah. Why do we call something a skin cell and not a gut cell and not a neuron? So cells have an identity, and the way in which cells express, it's thought that the biochemical basis of that is the way in which genes are switched on and off. That's what makes the cell type. There are certain proteins in some cells and not in others. The question is, what encodes that? How is that encoded? And it's not fully decodable from the linear sequence of genes, because it turns out that the way DNA is packaged in the genome and the modifications of DNA bases within the genome, they influence how genes are switched on and off. That's a field called epigenetics. And epigenetics probably has a profound contribution to make in understanding why it is that mutations happen in certain regions, certain tissues and not others, and why do some mutations have a different effect on a cell of a different type.
Okay. Thank you very much.
Yeah.
Thank you. Heck.
Yes, thank you. If I understood you correctly, what I've learned today is that within a cancer, if you look at the single cell level, there are different DNA sequences and mutations in those single cells. And I'm wondering if there are patterns or specifics that can be found that will predict which of those cells will be the fittest and or which will metastasize and which therefore should be the primary targets for new therapies.
Yeah, that's a great question. So that is in fact the subject of some of our research. And it's a difficult problem because what you want to be able to do is to say that this or that variation in the DNA is associated with a characteristic. It might be metastasis, it might be drug resistance. It's still a bit laborious to do. We can actually now do those studies because we can isolate individual cells and find the variation. We couldn't even do that 15 years ago. But the process of actually learning about which variant is strongly associated with a fitness effect, that's still a bit slow going. But I think that one of the openings actually may come from being able to sample DNA from plasma from patients that have cancers, because we're able in that situation to take frequent time interval samplings and then use the mathematics of population genetics, which turns out to be very powerful, in order to mathematically calculate what is the fitness of a particular variant. And I think in this way we're beginning to learn. We know for some very high fitness effect variants, there are some mutations which, when they happen, they're definitely going to confer a drug resistance. That's known to some degree. But it's the stuff in the middle that really accounts for a lot of the drug resistance that I think we're excited to be able to look into this. It's kind of an active area of investigation.
Across different kinds of cancers, you started to see similarities, for example, in what would lead to metastases, not just within a single type of cancer, but maybe there's a commonality that's shared across all cancers that can be exploited maybe 100 years from now.
Right. One of the fascinating things about cancer biology has been the discovery that there's only so many functions that are encoded in a cell. And we know that corruption of some of those functions will lead to the properties of a cancer. What's been fascinating is understanding that the route by which different cancers could get to that state is quite different. There are actually a multitude of paths by which you can end up in the same state. And that's part of the complication, is then trying to learn which confluence of factors might actually lead to this. I think we're heading in a direction where if we can anticipate what might develop, one might think about intervention in a way that blocks that particular path and blocks it from ever occurring. I think that's the question: is how do you defeat evolution? Yeah.
Okay, I'll be right back. I just want to ask a very highly voted question you have on Slido. So what challenges do you face when translating genomic discoveries in cancer into clinical application?
Oh, well, there's a free buffet out there. This is okay, what challenges do we face? Obtaining the knowledge is hard. I think translating it is in some sense even harder because research scientists don't control the domain in which translation happens. We influence the domain of translation by generating knowledge and pushing things forward, but we don't control healthcare policy, we don't control drug development agencies, we don't control pharma companies, all of these other elements have to combine for translation to happen. And translation has to be not just feasible, it has to be affordable. A big problem for our societies is if we develop interventions that we can't afford or sustain, then they aren't interventions. Now, eventually, technology and intervention can become commoditized if it's good enough. It will become affordable, but at the beginning, it doesn't look like that. And that's a challenge. That is a real challenge.
Thank you.
We could go on all night about that.
Over to you.
Thank you very much. First of all, that was great, well-developed speech. Yeah, I really used it. The question is, genome decoding will be copied or transferred to the next generation? And the side comment is that I'm a nature conservationist. And many of the natural, conventions at the international level, like UNCCD, CBD, or UNFCCC, all of them are talking about adaptation. And adaptation means that, the species that naturally adapted to the situation. I want to say that if we are changing the genome, will be a threat for the next generations or not. That's it. Thank you.
Oh, I get so. I want to see if I understood your question. If I understood correctly, you're asking, are we running risks by modifying genomes?
Exactly.
For the next generation?
Yeah.
So I'm not aware yet of anyone proposing. There's no reason in principle why someone wouldn't propose to modify a germline genome for some cancer prevention purpose. For example, hereditary mutations that predispose to cancer in principle might be correctable in the germline. And that's got huge ethical as well as maybe biological implications associated with it. I think somatic genetics is different. So here we're talking about not transmissible to the next generation, but rather transmissible only within the cells of the individual that's being treated. I think that's a different undertaking. It has technological issues, but I don't think it carries with it the same risk that you're discussing. Yeah.
Thank you. That was a great clarification. So we have another question, which is actually a couple of very good questions. Could you give a glimpse in how your day-to-day career and job might look like? What would a career in research entail? This might be from a student.
Okay. A day in the life of. Okay. I think the biggest misconception about research is the idea that we all sit in an office somewhere and secrete thoughts. Okay, it's not how research really works. You have to be passionate about the idea that you want to discover how something works or make progress in an area, because you're going to need to be sustained in that passion for all the other things that one needs, which is to be entrepreneurial. So you have to raise money. We probably spend about 50% to 60% of our time just researching funding or reporting on funding. That's another issue I'll mention, is that there's a whole sub-industry of report. Of course, everyone wants to know about the progress. But the more there is, the more has to be reported. So stuff needs to be reported. So you have to be entrepreneurial. You have to know how to recruit people, because you're not going to do research just on your own. You're going to recruit co-workers and trainees. They have to be trained. You need a lab environment. It's the closest thing to having a startup company than I can think of. I had people join my lab who have worked in startup companies and just said, wow, this is like being in a lab is like being in a startup kind of small company. And it is just like that. If you're not pushing it, no one else is pushing it for you. But the rewards are enormous, because you get to choose what you want to work on. And that kind of freedom is a very powerful thing.
Thank you. We've got 3 minutes left, and we've got 3 questions, so I think we can do it. Can genome sequencing help detect hereditary mutation early enough to prevent cancer? I think this is a good one.
Yes, and in fact it already does. British Columbia has a hereditary cancer program. Genome sequencing is a component of that kind of program, and it's predicated on the idea that individuals who have a family history, or may for some other reason suspect they could be at risk of having a cancer risk increasing germline variant, can have their genome sequenced after being interviewed and counseled by genetic counselors. So that's already happening.
So Sam, have you got your genome sequenced? Or should we all get ours sequenced? What are the pros and cons?
Yeah, I think, the question of pros and cons are, I haven't actually, and maybe I should. I'm pretty sure I know I have 1 or 2 alleles of something. Okay. The pros and cons, I think there's been a lot of public discussion. Obviously, if you sequence your genome, and there are all kinds of ethical implications. You might learn about family relationships you didn't know you had. You might learn about something that has a consequence for a sibling or another family member by virtue of heredity. In some jurisdictions, I think not in Canada, and actually probably not in the US and not in the UK, but maybe in some jurisdictions it might lead to people having difficulty with health insurance, for example. So there are downside implications as well as plus side.
Okay, so we have one more question, and we'll pick the top one then. Would baseline genome sequencing as part of routine healthcare set patients and healthcare providers up to better detect and respond to cancer?
As routine healthcare. I think that routine in cancer, if we refer to the genome as the genome of the cancer, I think that it would create a lot of benefits. There would also be a large number of patients who wouldn't be immediately helped. But the question is, what's the cost versus the downstream value of the intervention? That's something that I think hasn't been modeled out properly, because we know that treatments change, the cancers change. In a sense, it's like saying, well, we do histology on cancers to find out what type they are. That's used for treatment. Increasingly, genomics is used for treatment, and an increasing proportion of patients will benefit from some kind of genomic information. Where does the cost benefit sit in terms of doing that on everybody? Once it's cheap enough, and the cost of sequencing a genome is now well below the cost of an average imaging assay, an imaging study that's done in the clinic. Perhaps it would, and it would be, the counter arguments are sort of economic about the people who wouldn't immediately benefit, but I think a lot would, actually.
Well, thank you very much. I think we have finished. You want me to do the last one? Wait. Oh, of course. Yeah, that's a good one. What advice would you give young researchers entering the field of cancer genomics today? That's a good one to end with.
Right. What advice would I give people? Make sure you take some quantitative classes, because statistics and computation is kind of key to genomics. But I'd say, find a problem that really interests you, and there's no shortage of problems. There's been tremendous progress, as I tried to outline, but this story is far, far from written, and there's a tremendous medical need for progress in cancer. We have not fixed the entire problem. So find a problem that really excites you and just go for it.
Thank you. Okay, so I just would like to make an introduction now, ladies and gentlemen. Please, I would like to ask a Genome BC past chair of the board, Dr. Peggy Johnston, to come on the stage.
Thank you, Federica. And thank you, Sam, for a very tremendous presentation. As a cancer survivor myself, I really enjoy hearing and being inspired by the work that you and your colleagues are doing. As a gesture of our appreciation for sharing your insights today, I want to present you with this talking stick, which is hand-carved by the Coast Salish and Musqueam artist Wesley Wise. That's a picture. You can see it, yeah. The talking stick is used by many indigenous cultures and is an ancient and very effective communications tool. The person who holds the stick has the floor and is therefore able to share their wisdom, experience, and perspectives. And when that person is finished, the stick can be passed to others who can do likewise. This ancient tradition embodies the principles of equality, ensuring that every voice is heard and respected in the communal circle. In gifting this talking stick to you, we honor your presence with us tonight and the wisdom, experiences, and perspectives that you have shared with us, as well as your contributions to healthcare, science, and the well-being of people in British Columbia.
Thank you.
Before we wrap up the formal portion of today's organization, I want to give special thanks to several people. First, Dr. Federica De Palma for helping shape this year's event and facilitating tonight's Q&A session. And also a shout out to Sally Greenwood and our communications and societal engagement team, as well as all the volunteers who put together the various pieces that make this kind of event successful for not only today's activities, but also their outreach activities throughout the year. And so I'm going to conclude tonight's keynote portion of the program. We encourage you all to continue to discussion We're going to host a reception with light snacks and refreshments in the outside area. And please remember that all the Genome British Columbia staff have these green lanyards on. We invite you to engage with us, communicate with each other, and have a good time this evening. Thank you for coming and attending, and good night, everyone.